Hi, I'm Jim Watson, Mayor of the City of Ottawa. Thank you for joining and taking part in this year's Brain Health Awareness Week on November 8th to 12th, which I was pleased to proclaim. I'm excited to announce that next year's Brain Health Awareness Week will be taking place on October 24th to the 28th, 2022. I hope that you will join the U-Ottawa Brain and Mind Research Institute in this milestone event as the Institute celebrates its 10th anniversary in supporting world-class research on brain and mind-related health. As part of the week's events, the Institute is hosting a joint session with U-Ottawa Heart Institute called Discovering the Brain and Mind Connection, an exciting new collaboration in Ottawa. More to come in 2022. Thank you very much, and we wish you the very best.
Thank you, Mayor Watson, for closing Brain Health Awareness Week, and we look forward to next year's event. So good evening, everyone. Thank you for joining us. Bienvenue à toutes et à tous. Again, I'm Ruth Slack, and I'd like to welcome you to the final session of Brain Health Awareness Week that's hosted by the University of Ottawa Brain and Mind Research Institute. And this evening's program provides the opportunity to meet and celebrate our future generation of researchers. Tonight, we would also like to take this opportunity to thank and recognize our donors who support scholarships and fellowships at the University of Ottawa Brain and Mind Research Institute, which is essential to enable our trainees to pursue their research programs. So the mission of the University of Ottawa Brain and Mind Research Institute is to drive multidisciplinary research to accelerate discovery and innovation. But another part of our mission is to train and inspire the next generation of researchers. And I would say that this is really the most important part of our mission. Without a strong and inspired next generation of researchers, where would Canada be in the future? Where would medical research be in the future? So research is exciting and rewarding, but it can also be challenging. Science is intellectually complex. The lab work takes commitment, collaboration, and perseverance. And this is critically important for these discoveries in research to materialize. And it's a critical part of the learning process. So tonight we are delighted to celebrate the accomplishments of our research trainees, including the recipients of our University of Ottawa Brain and Mind Research Scholarships and Fellowships. And once again, I'd like to express our sincere gratitude to our donors who provide this critical scholarship support for our trainees. And now it's a pleasure to turn this event over to Teresa Chan, who will speak on behalf of the trainee committee. Thank you, Teresa.
Thank you, Ruth. Hi, everyone. My name is Therese. I'm a member of this year's Brain Health Awareness Week Planning Committee, and I will be one of your co-hosts for tonight's program. I'm a second-year master's student in the epidemiology program at the University of Ottawa. I'm co-supervised by Dr. Doug Manuel and Dr. Amy Hsu, and my research focuses on looking at whether different ways of measuring hearing impairment can improve a risk prediction model for dementia. This evening, we have an exciting program to share with you that will be recognizing individuals for their research accomplishments, as well as acknowledging the donors who have kindly supported research scholarships and fellowships for the 2020 to 2021 year. I would now like to introduce Michaela Lunn, a fellow member of the Brain Health Awareness Week Planning Committee. Michaela will be introducing the first group of awardees that will be presenting this evening.
Hi, like she mentioned, my name is Michaela. I'm a third-year PhD student in the lab of Dr. Michael Schlossmacher. I study the role of the immune system in the onset of Parkinson's disease. And as a previous PRC recipient, I am very excited to introduce the highly talented awardees of this year. I would like to mention to the attendees that if you have any questions while the event is happening, you can put it in the Q&A box, and we will hopefully be able to answer as many as we can at the end of the event during the Q&A session. So first, I'm happy to introduce Kareem Ibrahim. He is a PhD student in the lab of Dr. Stephen Ferguson at the Faculty of Medicine at UO. He was awarded the Mark and Gail Markoglias Graduate Fellowship, and he is studying the role of vesicular glutamate transporter VGlut3 in Huntington disease. Unfortunately, Kareem could not make it tonight, but he did pre-record a video for us, which I will share for you now.
Good afternoon. My name is Kareem Ibrahim. I'm a PhD candidate in the Dr. Stephen Ferguson lab. I'm very excited to take part in this conference, and I would like to thank all the organizers and volunteers for putting such huge effort for this event. In the next few moments, I will briefly walk you through my research project, which is focused on studying the role of vesicular glutamate transporter 3, or VGlut3, in Huntington disease. So a little bit of introduction about Huntington. Huntington disease is a genetic fatal neurodegenerative disorder. It's caused by mutation in Huntington gene, which leads to accumulation of abnormal form of this protein or mutant Huntington. This leads to degeneration of the brain cells, as you can see here, over the course of a period of years. Huntington disease is a relatively rare disorder. In Canada, it's around 17 per 100,000 Canadians have Huntington disease, with 3,700 more of high risk of developing the disease. The symptoms are more or less similar to Parkinson's disease, where you see movement abnormalities or jerky movements such as chorea here in the picture. Also, you see a progressive decline in cognition and dementia, and some psychiatric symptoms such as depression and anxiety. And although the nature and the pathology of this disease is very well studied, till now, there are no curative therapies for patients, only medications that can manage the symptoms. So looking closely into the causes of degeneration Huntington disease, one particular region is severely affected, and this region is called the striatum. The striatum is important for coordination of body movements, and the cells of the striatum were found to be more sensitive to glutamate. Glutamate is one of the major neurotransmitter molecules in the brain cells, which they use for communications and to mediate their activation and excitation. Overall, this hypersensitivity leads to neuronal overexcitation or excitotoxicity. And one of the early studies in the field in baboons or primate models of the disease, where they injected an activator of glutamate receptors, ibofluronic acid. And this activator caused an evident lesions in the striatum, as you can see here on the right side, compared to the non-controlled or non-treated side of the striatum. Also, in mouse model of Huntington's disease, when we treat the cells with glutamate, this increases the number of apoptotic cells or the dead cells here in the green spots compared to the non-diseased mice. Also, our lab previously showed that if in striatal cell culture of Huntington's disease, when we treat with a glutamate activator DHPG, we see a spike or increase in calcium signals. And this increase in calcium signals means that the cells are hyper-excitable than normal, which ultimately accelerates the death. Therefore, glutamate signaling is an interesting target in Huntington's disease pathology, and there's a lot of study in the field, including from our own lab, showing that glutamate receptor modulators have promising therapeutic potential. My research is focused on VGLU3 or vesicular glutamate transporter 3. And simply, this protein is involved in the packaging of glutamate and the presynaptic vesicles of the neurons or the brain cells. Therefore, they can indirectly affect glutamate release. What's unique about VGLU3 is that it's expressed in discrete regions within the brain, including the striatum, and of course, they are expressed in the hippocampus, dorsal raphe, and they are scattered within the cortex. In these regions, VGLU3 fine-tunes the co-release neurotransmitters, including acetylcholine, serotonin, or GABA, which means that VGLU3 can be an important regulator of regional brain network activities. Recent studies have shown that VGLU3 signaling exert glutamate-dependent currents on the spiny neurons, which are the main cells of the striatum, and also on the interneurons. As you can see here, when we delete VGLU3, it suppresses or decreases the excitatory currents on the interneurons, and also the same case in the medium spiny neurons. Other studies have shown that VGLU3 also appear to mediate the brain functions which are involved in the locomotor behavior in animals, and also in response to cyclostimulants. However, evidence on the role of VGLU3 in Huntington's disease is still lacking. So the first part of my project attempted to answer this question. Does VGLU3 play a role in Huntington's disease progression? For this reason, I used mice where VGLU3 gene is deleted in both non-diseased and diseased Huntington's disease mice, which express the mutant Huntington protein. I did behavior assessment for these mice from 16 to 15 months of age because the Huntington's disease mice start showing the symptoms around 9 to 10 months of age. For assessment of cognitive and short memory function, I use novel object recognition tests, while for motor coordination and motor function, I used rod, horizontal ladder, and grip strength test. In novel object recognition tests, it's simply a 2-day test. In day 1, we place the mouse in a box to explore 2 objects, A and B, and they are similar. And we calculate the time for them to explore these objects. And as you can see here, in male and female mice, all of them exhibited similar exploration time recognition index toward object A and B, which means that they do not discriminate or they do not have preference towards these objects. On day 2, however, I switch one of the familiar objects with a novel one, and if the mouse recognizes this familiar object, it will spend more time exploring the novel object. And at 6 months of age, we find that deletion of VGLU3 in males and females doesn't affect their memory. As you can see here, it shows high recognition index toward the novel object. And also we see the same case for the Huntington's disease mice, which is expected because this is before the typical age to show the symptoms. At 9 months of age, again, we didn't see any impairments in memory, as all of the tested mice, males and females, showed high recognition index for the novel object, which denotes that cognitive functions are still intact at this age. However, at 12 months of age, we can start to see that Huntington's disease mice show dysfunction in memory. They couldn't differentiate between the familiar and the novel objects, both in male and female mice. And also VGLU3 loss didn't rescue such impairments in Huntington mice, either in males or females. Interestingly, in 15-month time point, we see that VGLU3 loss in Huntington male mice improved the recognition index for the novel object. However, we didn't see such a similar case for female mice, suggesting that VGLU3 might play a role in memory dysfunction Huntington disease in a such sex-specific manner. Moving to rotor-rod test. In this test, we place the mice on accelerating rotor rod, and we calculate the time for them to fall off. So if the mouse stays longer on the rod, it means that it has intact limb coordination or movement coordination. In main animals, you see here the male Huntington male mice, the solid blue line, showed persistent decline in the latency to full periods, starting from 9 to 15 months of age. However, deletion of VGLU3 rescued the latency to full values up to 15 months time point to similar levels to the wild type or non-diseased animals. We see similar observation in female mice as well, where there is significant and progressive decrease in latency to full period, which is rescued by VGLU3 loss in these animals. Third test is horizontal ladder test, in which we assess the mouse limbs coordination by counting the number of missed steps while crossing a ladder with irregularly spaced steps. Similar to rotor-rod test, we can see that both male and female Huntington disease mouse have higher percentage of missed steps or percentage errors from 9 to 15 month time points. On the other hand, loss of VGLU3 significantly decreased those values to comparable levels to the non-diseased mice of both sexes. In the last test, grip strength test, in which we assessed the mouse forepaw grip, we find that the loss of VGLU3 rescued the impairments in Huntington disease animal at 9-month time point, both males and females. However, after that, they were not significantly different from the Huntington animals. And this suggests that VGLU3 rescue is more evident in motor learning and coordination rather than muscle strength. So in summary, I showed you that VGLU3 loss led in Huntington disease animal led to sex-dependent improvement in novelty memory deficits, particularly in male mice, and loss of VGLU3 offered long-term improvement in the Huntington disease motor coordination deficits throughout 15 months of age. Overall, VGLU3 represents an interesting target for Huntington disease, and understanding its role in disease pathology will definitely provide reasonable ground for tailoring selective VGLU3 modulators to be used in therapeutic management for Huntington. The study is still ongoing, and we are now looking for how VGLU3 loss influence the disease pathology at the molecular level. With that, I would like to thank my supervisor, Dr. Steve Ferguson, for everything, and all of the lab members, especially Khalid, for his continuous support and guidance. And also, I'm grateful for my committee members, Dr. Slack, Dr. Albert, and Dr. Tiberi for their support, and of course, for the funding agencies and Mr. and Mrs. Marco Glazy for funding my graduate fellowship. Thank you.
Thank you, Kareem. I'm sorry, I was having trouble starting my video. Thank you, Kareem, for sending us that pre-recorded video. And next, I am happy to introduce Margarita Louis. She is a third-year master's student in Dr. Jing Wang's lab. She is in the Faculty of Medicine at U Ottawa and part of the Ottawa Hospital Research Institute. And she is a recipient of a trainee scholarship for stroke research. Margarita is exploring regenerative strategies for stroke and neurodegenerative diseases. Welcome, Margarita.
Thanks, Michaela, for that introduction. So again, my name's Margarita, and today I'll be sharing with you a bit about my project and specifically some of the techniques that I use to basically explore some novel stroke therapies that are in development. So firstly, I'm in particular interested in these cells called pericytes. So pericytes are closely associated with the vascular system, and you can find them basically lining the blood vessels. And here they function to help regulate blood flow as well as the development of new blood vessels. And so in the brain, they can also be found interacting with astrocytes and neurons and still wrapping around the blood vessels that are found there. So an example of what these look like in actual tissue. So the red is the blood vessels, and then the green is the pericytes laying closely next to them. So my project, there's something special about these pericytes, which is why I'm studying them, and that is that when they're exposed to ischemic conditions, so basically deprivation of oxygen and nutrient support, they actually undergo this reprogramming process into neural stem cells. And then these neural stem cells can go on to differentiate into mature neurons. And so from here, we basically have this local source of cells that can supply stem cell pools to help replace the injured neurons that are found at the stroke lesion. And so in our lab, we've actually found a pathway that underlies this whole reprogramming and differentiation process. And so basically when this pathway is turned off, we see an enhancement of reprogramming. And then when it's turned on, we see an enhancement of differentiation. And so we've also conducted a variety of studies and found specific drugs that can turn on and off this pathway. So specifically, we have compound C that can turn off this pathway and boost reprogramming, and then JZL 184, which can turn on this pathway and boost differentiation. So for my project, we developed this combination drug treatment in which I first administer this compound C to enhance reprogramming, and then follow that up with JZL 184 to enhance differentiation. And so to test this in actual and its effects on actual stroke, we use stroke modeling in animals as well as behavior testing to study stroke outcomes, recovery, and also drug effects, which I will be going into more later in this presentation. So firstly, for our animal stroke model, we do a procedure that's similar to what we do for brain surgery in humans as well. So they're set up in this stereotaxic frame that allows us to target specific brain regions. And we administer a small amount of a combination of vasoconstrictors, basically to stop blood flow to the area. And those are called ET1 and LNAME. And so we specifically target the left side. And so on the brain, mouse brain specifically here, we see, we actually target the left sensory motor cortex. So we have some measurable outcomes of our stroke. So to introduce a bit of terminology, so the side that is injured is referred to as the ipsilateral, or the same side as the injury, and then the side opposite to it is called the contralateral side. And you'll hear me refer to these two terms throughout this presentation. And so in terms of actual testable outcomes, so if we have our cortical injury on the left side, then much like in humans, we see this motor impairment manifesting on the opposite right side. And so when we go and do our motor behavior tests, we have to do first a baseline to see how well these mice perform naturally. And then after they undergo this stroke procedure, They're tested again in my timeline, at least, at 7 days, 14 days, and then finally 35 days following the initial stroke procedure. And so since I'm testing a drug combination, I'm comparing two groups of mice. So one that receives just saline treatments, and then one that receives my proposed drug combination paradigm of compound C followed by JZL. And so now we'll get into the actual motor tests, which Kareem also mentioned earlier in his presentation, but I'll be going into more depth with you guys here. So the horizontal ladder test, as he showed us, looks a bit like this. And the mouse just has to make it across from one end to the other, a bit like an agility course. And so the purpose of this is to test the mouse's motor coordination. And so at baseline, so no injury, the mouse actually has a training day first. So it gets to know how to cross this ladder without any stopping. So at baseline, we should see only minimal errors, which we define as any slips or misses between the rungs. And when it grasps the rungs to cross, they should be pretty neat, and their fingers should wrap around the bars. And so again, referring to this, where we'll have our right impaired side. So following stroke, the contralateral right pause will be more prone to error. And so this again is our horizontal ladder. And specifically, our setup has uneven rungs. So at the starting end, the rungs are more evenly spaced. So it's a little bit easier for the mouse to predict where to place its paws and cross. And then at our end, as we go from start to end, it actually gets more uneven. And so it becomes more difficult for the mouse to maneuver across this without making any mistakes. So I'll actually show you what this looks like. And we always analyze our video footage at very slow speed. So specifically, so I can show you what this looks like. We're going at 0.06 times speed of the original footage. And so again, this is the starting end with the even rungs. And then at our ending side, we have the more uneven rungs. And so when we're looking at this, our left paw will be placed, if the mouse is coming from this end, it'll be placed on the far side of the ladder in the video. And then our contralateral injured or to be injured side, right side will be placed closer to us in the footage. So we'll just look at that for a minute. And so there's left, and then there's right, left, right. And so we'll do this a couple times so we get enough footage. And for our lab, we sample 3 trials and then average their performance across. So as you can see, there's no slipping, and almost all of the paws are grasping the rungs very neatly and not just catching on its elbow or anything like that, or its palms. Okay. And then so following stroke, so what we actually do is from week to week, we'll actually flip the ladder around. So this is specifically day 7. So instead of them starting on this side, our starting end is actually on the right side. And then our end is on the left side now. So the mouse will be coming from this side. And then with that, where we're looking at the positioning of the paws also switches. So left side is now closer to us, whereas the now injured contralateral side paws will be further away from us in the video. And so we'll take a look at that now. Again, coming from this end. So there's the right paw being placed, and then the left paw. There's the right slipping, and then again, that right hind slipping. So left, right, just missed. And then left front, right front, left front, right front, left front, and then that right again is missing that coordinated movement. And then again, right back, just missed that one as well. And so we can clearly see the effects of our stroke procedure on this coordinated movement compared to our pre-stroke baseline. Okay, and then basically it's over when the mouse's hind paws cross that little tape. And so in terms of results for my project, so the treated mice contralateral side is what we're going to be looking at mainly. So ipsilateral is basically 0% error. So contralateral, we can see actually at day 7, they both have pretty similar percentage error. But then at contralateral, we see greater improvement or return to baseline performance in our compound C and JCL treated mice as compared to our saline treated mice. Okay, so next will be a test that Kareem didn't mention before. So this one is called the cylinder test. And so this actually tests paw preference during rears. So the setup looks a bit like this. So we have this transparent cylinder that the mouse is placed in, and then we have an overhead camera to film a bunch of rears that the mouse will do when it's placed inside. And so a rear is basically when it stands up on its 2 hind feet. And then what we're looking at is basically these forepaws. It'll reach out onto the sides of the glass cylinder to balance itself. So at baseline, the mouse typically doesn't have any strong preference for either left or right paw. But then following stroke, with again, our right side being the impaired or injured side, since we have left side injury, cortical injury specifically. So after stroke, it'll actually show a greater preference of using that ipsilateral left paw because that contralateral right paw is impaired. So I'll go and show how the baseline performance looks like first. So this one's at regular speed, and you can see he's using pretty evenly both sides when it stands up like that. Both sides. And then post-stroke, we can play this too, and then you'll see left side preference for this. So again, using that left paw to balance more so than on the right paw that you are seeing it using in the baseline. And so for my project results for this, we actually see that they are still equally as different from baseline, but there's less ipsilateral paw preference throughout for the compound C, and they have a greater return to baseline preference in the drug-treated group. So those are some encouraging results, still probably including some tweaks here and there to better optimize this effect, but that's basically all I'll be showing today. So thank you guys for listening, and I hope you enjoyed that. Big thank you to obviously my lab, the Jing Wang lab, and all its members, and then particularly the Behavior and Physiology Core, who allows us to do all of this animal work with the testing, supplying us all the things we need to do it, and then ACVS for providing all the care for the animals and ensuring that everything meets the regulations, and as well to our donors, finally, for your generosity and support, without which we couldn't continue to do this exciting and important research.
Thank you, Margarita. Next, I will be introducing Brian Doris. He was awarded the Trainee Scholarship for Stroke Research as well. He is supervised by Dr. Zodong Chow, and he's currently a master's student of Applied Science in Biomedical Engineering at U Ottawa. His research interest revolves around studies that transition extracellular vesicles from bench to bedside. So I'll now welcome Brian.
Hi, everyone. So my presentation will focus on developing an extracellular vesicle releasing duroplasty for stroke treatment. So stroke can be categorized as either hemorrhagic or ischemic, where the latter occurs when a blood clot blocks or narrows an artery, obstructing blood flow to other brain cells. Stroke has become the third leading cause of death in Canada, and many survivors are left with one form of disability that might require intensive rehabilitation. Furthermore, there are a few patients that can experience brain swelling during an ischemic stroke that result in increase in intracranial pressure, leading to damaging even more brain tissue. One of the most effective treatments for brain swelling is performing a decompressive craniectomy, where a portion of the skull is removed and the dura is opened to expose the brain. Then, to protect the brain from the environment while still allowing the swelling to occur outside the skull, a duroplasty is placed over the opening. This procedure is shown to reduce mortality rates, but most are still left with some form of a disability. Therefore, our overall project goal is to develop a duroplasty that can directly deliver a therapeutic treatment to the brain for possible functional recovery. We want to utilize the regenerative and immunomodulatory properties of stem cells, in particular mesenchymal stem cells, or MSCs, to promote the functional recovery of stroke patients. So MSCs are adult stem cells that can be isolated from bone marrow, cord blood, adipose tissue, or umbilical cord tissue. MSCs have been shown to promote endogenous neurogenesis and angiogenesis by releasing specific trophic factors, and can regulate the immune system post-stroke by preventing the activation and infiltration of immune cells in the brain. As a result, MSC treatment has shown to promote functional recovery within rats, and one of the most important factors released by MSCs that contribute to functional recovery are extracellular vesicles, or EVs. EVs are spherical, heterogeneous lipid bilayer capsules that are released by most cell types that can have a diameter ranging from 50 to 2,000 nanometers. They have shown to have similar therapeutic effects for treating stroke as their parent cells due to their content having similar trophic factors and microRNA that promote stroke recovery. A very important characteristic that MSC EVs inherit from their parent cells is their allogenicity, meaning a patient can be treated with MSC EVs derived and collected from multiple healthy donors without experiencing any immune reaction. So therefore, our research involves developing a method to directly deliver MSC EVs to the stroke brain. The first step of our project is determining optimized storage conditions for these EVs so that we have enough readily available EVs once treatment is needed. Then this will be followed up by loading our cellulose-based duroplasty with EVs and determining its release profile. We will then observe whether the EV-loaded duroplasty will promote neurogenesis and angiogenesis in vitro. Then, to see if functional recovery is observed in vivo, a stroke is induced in rats and a decompressive craniectomy is performed to load our EV-loaded duroplasty. My research will focus on the optimization and storage conditions of EVs. Then, as for collecting and isolating EVs, we have partnered with the Stem Cell-Based Therapeutics Laboratory Regulatory Research Division of Health Canada to receive regular shipments of MSC EVs. So storing freshly isolated EVs at 4 or negative 20 degrees Celsius results in a loss of more than 50% of its content, while storing at negative 80 for the same time period did maintain its physical and functional properties, but past 30 days, particle and protein concentrations starting to decrease. Therefore, to ensure a large amount of EVs are quickly ready once needed, freeze drying, or also known as lyophilization, can be done to store EVs at room temperature for an extended period of time. Furthermore, our duroplasty, like the ones that are commercially available, will also be freeze dried, allowing us to incorporate the EVs with our duroplasty. However, freeze drying EVs still result in a loss in some particle concentration, protein concentration, and some EVs can start to aggregate. Then the drop in particle concentration occurs during the drying process as water molecules that interact with the lipid bilayer are removed and the membrane becomes disordered and breaks apart. Therefore, to help reduce the decrease in particle size, a chiroprotectant is added to essentially replace water during the freeze drying process. This is known as water replacement theory, where once water is removed, trihalose will then interact with the lipid bilayer to ensure that the lipids maintain their structure. In a similar fashion to protecting the lipid bilayer, trihalose is then theorized to diffuse through the lipid bilayer of the EVs and then interact with the content within to protect it during the freeze drying process. So to optimize the freeze drying conditions, we decided to test multiple trihalose concentrations at different freezing temperatures while storing the samples for 2 weeks at room temperature. My current tests will test all the desired trihalose concentration at a freezing temperature of -80. We then characterize EVs via nanocyte to determine the size distribution, particle concentration, and particle characteristics, and perform a BCA assay to determine the protein concentration post-freeze drying. So freeze drying EVs without any trihalose results in a decrease in particle concentration and a wide distribution, indicating aggregation as seen with the orange curve, which is what we expected. However, even when we add around 10 millimolars of trihalose, similar results are observed. This can be due to there not being enough trihalose to incorporate into a majority of EVs to protect them during the freeze drying process. The wider size distribution can also be emphasized by the increase in mean diameter due to EVs aggregating, as we see the green bars indicating mean diameter are increasing after the freeze drying process. However, as we increase the trihalose concentration from 0 to 200 millimolars, so from the orange to red to purple and then to the blue curve, we can start to see that the size distribution becomes narrow and only one peak is observed, indicating less aggregation and more particles are recovered. At a concentration of 200 millimolars, we even see more particles are recovered, which is what we want, suggesting that trihalose is incorporated into the EVs. However, trihalose, even with the amount of trihalose we add, there's still some loss in EVs and increased size in particles. The loss of particles could happen due to osmotic shock that causes the EVs to burst, or EVs could still be aggregating together slightly, and when nanocyte measurement is performed, the machine could analyze a clump of EVs as one particle rather than a group of particles. The increase in average diameter, the green bars, also suggest aggregation is occurring. Then we analyze the protein concentration post-freeze drying. So even with 10 millimolars of trihalose, the black bars still resulted in reduced protein concentration, which can be due to not enough trihalose being added as well. Furthermore, the drop in protein content might also have to do with not enough water being removed in combination with the lack of trihalose. So to ensure that a sample is to be stored for a long period of time, the goal of freeze drying is to remove as much water as possible. And if the water content is greater than 1%, the sample will have a lower shelf life at room temperature. However, reducing the water content below 1% can be very difficult, as over drying the sample can also start to degrade your protein. Furthermore, a BCA assay involves a reaction of multiple peptide bonds of a protein with BCAA sodium salt to induce a color change in a sample to indirectly determine the protein concentration. However, when the residual moisture is greater than 1%, water molecules can react with the peptide bond via a hydrolysis reaction during storage to break down the protein chain and will not be involved in the BCA action, resulting in a reduced protein concentration. However, when we increase the trihalose concentration, there's a possibility that trihalose molecules are diffusing through the membrane due to a large enough concentration gradient being formed to form hydrogen bonds with the peptide backbone of the protein to protect it from the hydrolysis during storage, as you see an increase in protein concentration post-freeze drying. So even with the results we have, there's still experiments that need to be done to verify that our EVs retain their therapeutic capabilities post-freeze drying. To ensure that our results are reproducible and the best freeze drying conditions are determined, we'll have to continue repeating these experiments multiple times. Then to observe that our MSC EVs retain their angiogenic capabilities, a tube formation assay will also be performed. We will also co-culture these EVs with neuronal stem cells to see if they promote their proliferation and growth. Then to determine the MSC-EV release profile from the duroplasty to gain a better understanding of what the protein and particle concentration is as EVs are released over time to ensure that a therapeutic dose is reached for functional recovery. So thank you everyone for listening. I'd like to thank my supervisor, Dr. Zuan Chow, and everyone at our laboratory. And I'd like to thank Dr. Jessie Lavoie at Health Canada for providing us with our MSc EVs to do this study.
Thank you very much, Brian. Great talk. Next, I'm going to introduce Zoe Fisk. She is the recipient of the Talk Family Fellowship. She is a second-year master's student in the lab of Dr. Maxime Rousseau at the University of Ottawa in the Department of Cellular and Molecular Medicine. Zoe is interested in studying the involvement of alpha-synuclein and Parkinson's disease, and today she will be showing us some of her project as well as a specific technique that helps her reach her research goals. Welcome, Zoe.
Thanks for the introduction, Michaela. All right. So yeah, like Michaela just said, my name is Zoe. I'm a second-year master's student under the supervision of Dr. Max Rousseau, and my project focuses on the role of alpha-synuclein in Parkinson's disease. So just a brief background on Parkinson's. I'm sure most people here have heard of it or have been affected by it in some way. It's actually the second most common neurodegenerative disease worldwide, and it presents with a multitude of motor and non-motor symptoms. So if you look at this diagram on the right here, I've outlined what some of these symptoms are. Most people, when they think of PD, they think of tremors and issues with walking, slowness, but there's also a lot of non-motor symptoms, including anxiety, cognitive decline, and GI issues that can come with the disease, and it leads to a very poor quality of life for those who have it. There's no cure for Parkinson's disease as of right now, and there's only treatments that affect the symptoms and not the pathology. So speaking of the pathology, there are two hallmarks of Parkinson's disease. The first of which I'm not going to focus on, but it's dopaminergic neuron loss in the substantia nigra, and this leads to a lot of the movement issues as there's reduced dopamine signaling in the brain. And the second hallmark, which is what my project focuses on, is the formation of Lewy bodies in Lewy neurites. So Lewy bodies, which I've shown on this diagram here on the left, are these clumps of aggregated protein that are composed of alpha-synuclein. So alpha-synuclein is this presynaptic protein. We don't really know a ton of its function. We know that it's somewhat involved in neurotransmission. However, its function does remain elusive. And in Parkinson's disease, this presynaptic cytoplasmic protein starts to aggregate and clump throughout a neuron. A little bit of it actually goes into the nucleus to cause toxicity. And as this aggregation and toxicity spreads, it leads to neurodegeneration and cell death, and then the formation of these Lewy bodies. So these Lewy bodies lead to all sorts of chemical imbalances and cause some of the symptoms of Parkinson's disease. So alpha-synuclein is really difficult to study in its native form, since it is a cytoplasmic protein. When you stain for alpha-synuclein using immunostaining techniques, it shows a really diffuse signal, and it doesn't give you very much information on the cells that this protein naturally occupies. So we don't know about its cell specificity. We also don't generally know its triggers of aggregation, so it's really important that we further study this protein to understand PD better. So to combat these issues with studying alpha-synuclein, my lab has generated the SNCA NLS mouse, and what this mouse model does is in the SNCA gene, we've inserted a nuclear localization signal, or NLS, which forces all cytoplasmic alpha-synuclein into the nuclei of cells. So I just have a little illustration of that on the left here, but on the right, you can see in a wild type mouse, when you stain for alpha-synuclein, you get this really diffuse signal that I mentioned earlier. You can't really see cell type specific staining, and it just looks like the protein is everywhere. However, in our mouse model, when you stain for alpha-synuclein, you get this distinct nuclear staining, and you can see exactly where this protein is. So both of these images have the same amount of alpha-synuclein. It's easier to visualize it using this mouse model. So my project focuses on what cells contain alpha-synuclein and if there's a pattern to alpha-synuclein expression. So this brings me to my project. And instead of just showing everyone my data, I actually want to walk through some of the techniques I use and how I go from getting my mouse tissue to getting my data. So the aim of my project was to generate an organism-wide atlas of alpha-synuclein topography and its cells of origin. And the steps that I'm going to be walking you guys through today are my tissue selection and sectioning processes, a staining method I use called immunohistochemistry, machine learning analysis that I use to actually quantify my data, and then another staining procedure called immunofluorescence. So the first thing I do when I get my mice is I actually collect brain and peripheral tissue from them. For this portion of my project, I've used 6 mice, and then I flash freeze that tissue. So on the left here, I actually have a flash frozen mouse brain. It's super tiny. You can see it in comparison to my finger there. But what I do is then I take this tissue that's frozen and I use a machine called a cryostat, which I have pictured on the right, to section this tissue super, super thin. So I actually section my tissue at 20 microns, which would be impossible to do by hand if you didn't have the help of a machine. So this is the cryostat. I accidentally cut it off in the image here, but on the right side of it, there's actually a wheel that you can crank and it just basically cuts the tissue. So what I do is I take my mouse tissue, embed it in this material called OCT, and then I can actually put it into the cryostat, which is on the right. And then I don't know if you guys can see my mouse, but this little black area here, there's a block. You put the tissue there, and then just below it, there's a very sharp blade that allows for sectioning of the tissue. So once I have my tissue, I mount it onto slides, and then I'm able to do immunohistochemistry or IHC to stain for the protein I want. So this is what the staining looks like. And I apologize because my handwriting is pretty terrible here, but these are what my brain sections look like after they've been stained. So this is what a coronal stain or section looks like. And then below that is a sagittal sectioned brain. And this is just so you can look at the brain from 2 different orientations, but everything indicated in brown here indicates a positive signal for alpha-synuclein once I've stained it. So the brain, if you considering this image here, you can see that the brain is white, but once I've stained it, it turns brown. So then I take this data, upload it using a slide scanner. Oh, here's also some peripheral organs. I have mouse kidneys here, and then the mouse small intestine, because I'm also looking at peripheral organs. And then what I do once I have scanned all of my images is I put them into a program called Elastic, which allows me to count exactly how many alpha-synuclein positive nuclei I can see with my stain. So this is what the interface looks like. These white arrows here indicate a positive alpha-synuclein signal, and then the black arrow indicates where I've labeled background in blue. And basically I train this program Elastic how to identify these 2 different regions through all of my brain sections. And after a couple of rounds of learning, it's able to process all of my slides. And this is what the output from Elastic looks like once I've played around with it using another program called Fiji. But basically every single red dot here indicates 1 positive alpha-synuclein cell. And using this, I was able to construct a whole brain-wide atlas showing exactly what cell types this protein is contained in. This is a very brief version of my atlas, the coronal section of the atlas. I've only included about 9 sections here, but there's actually, I believe, 162 different coronal levels that I could have shown. But you can see this is directly from the front of the brain, the olfactory bulb, all the way to the back where the cerebellum is. So I can see exactly where this protein is throughout the brain. Then to further confirm where alpha-synuclein is in the brain, I used another technique called immunofluorescence. And this staining method is a little bit different than IHC. You can actually stain for multiple different proteins at once, and you can use different antibodies to do this. And I also find it's a little bit more visually appealing than IHC. So you can see here in red, I have stained for alpha-synuclein, and you get that nuclear staining that happens in my mouse model. And then I've also stained for TH, which indicates dopamine neurons. So this is actually the substantia nigra, which is where the dopamine neuron loss occurs in the brain. And you can see co-localization of alpha-synuclein with these dopaminergic neurons. So as a whole, using my atlas, I was able to detect that alpha-synuclein primarily exists in granule, pyramidal, mitral, dopaminergic, and layer-specific cortical neurons. And this led me to believe that there might be a preference for alpha-synuclein to exist in excitatory neurons. And I'm really excited to explore this further with the rest of my research. So that's about it for my presentation today. I want to give a huge thank you to the Toth family for allowing me to work with them and giving me their fellowship last year. And I also want to thank my lab members who are, most of them are pictured on the right here.
Thank you so much, Zoe. And thank you to all of the speakers we've just had. I'm now going to pass this role over to Therese to introduce the remainder of the speakers.
Thank you, Michaela. I'm happy to introduce Dr. Paolo Polanco as our next presenter. Dr. Polanco is a radiologist and research fellow based at the U Ottawa Faculty of Medicine's Department of Radiology and is supervised by Dr. Richard Aviv. His research interests involve imaging techniques in stroke, brain tumors, and head and neck pathology. He was awarded the Michael T. Richard Fellowship and has a video presenting his funded research project using machine learning to predict hematoma expansion in ICH.
Hello everyone, my name is Paulo Polanco and I'm a research fellow working at the Department of Radiology at the Ottawa Hospital under the supervision of Dr. Richard Aviv. I want to start my presentation by thanking Dr. Michael Richard for the funding opportunity obtained this year. This research project primarily focuses on using machine learning, a type of artificial intelligence, to predict hematoma expansion in patients with an intracranial hemorrhage. So let's begin. There are two types of emergency related to blood vessels in the brain. The first one is an ischemic stroke, which is an occlusion of the main artery of the brain, resulting in an interruption of blood flow to an area of the brain that eventually can die if no intervention is performed. The other type of stroke, the focus of our research, is defined as a hemorrhagic stroke. This condition is secondary to the rupture of a blood vessel in the brain, resulting in bleeding extending into the brain parenchyma. The rupture of a vessel could be secondary to the presence of a pre-existing aneurysm, which is a focal dilatation of the vessel, or due to vessel rupture secondary to a high blood pressure. This is an example of a positive case. This is what we usually see in a patient with an acute cerebral bleed. This brain scan shows a parenchymal bleed, which is seen as this white stain in the brain. Since the brain is surrounded by bone, there is no room for any additional space-occupying lesions. That means that in the case of bleeding, a hematoma creates its space by pushing all the structures around it. Let's see another example. On the left side of the screen, we can see a normal brain with all the structures symmetrically and of normal size. Now, the patient on the right side of the screen has acute bleeding. We can see how the bleeding pushes everything and creates its own room. But the problem is that by doing this, it displaces normal structures towards the contralateral side, and this becomes a problem for the patient. Now that we know what a hematoma is in the brain, let's talk about hematoma expansion. In a general sense, a hematoma expansion is a growth in the hematoma volume over time. Here we can see the growth of the hematoma between the baseline study and the follow-up study obtained 24 hours later. So why does hematoma expansion matter? Well, hematoma expansion occurs in up to 40% of all intracranial hemorrhage patients and is well documented as a marker of poor prognosis and functional outcome after intracranial hemorrhage. Expansion of the initial hematoma is so far the only marker of outcome that is at least partially preventable. Thus, the prevention of hematoma expansion is an attractive target for the treatment of intracranial hemorrhage. Hematoma expansion predictors have a vital role to play in selecting high-risk patients and subsequently facilitating individualized treatment. The issue is, hematoma expansion treatment has to be delivered as soon as possible to limit expansion, but how do you know at baseline whether a patient will expand or not? For example, we can use clinical predictors of hematoma expansion. Many studies have used statistical models to try and identify associations between clinical features and hematoma expansion. One of these features is the use of blood thinners. These drugs have different mechanisms of action, but the end goal is to impede clotting. That means that if a vessel ruptures in a patient under treatment with any of these drugs, it is more difficult to stop the bleeding since these drugs prevent clot formation. Another essential clinical marker is blood pressure. You may ask why hypertension may lead to hematoma expansion. What can happen is that If you have high blood pressure, the force on your artery walls can lead to more bleeding into the brain. Furthermore, blood releases from the initial bleeding can put pressure on smaller vessels and lead to their rupturing, which can partially explain hematoma expansion. There are a lot of other clinical markers that people have identified, for example, clinical scales and laboratory findings, but we will not get into this today. Now let's switch gears into what we as radiologists like most, imaging studies. We're very creative and observant when dealing with abnormal findings in imaging studies. In addition to clinical markers, there are also radiological signs that can be predictive of hematoma expansion. We can classify the baseline bleed based on its appearance on the initial CT scan. We can assign a number on a 5-level scale, for example, as in this scenario, or we can try and identify several imaging signs such as the swirl sign, the presence of hypodensities within the hematoma, the black hole sign, and the blend sign. If any of these signs is present, that means that there is an increased risk of hematoma expansion. The problem with radiological signs is that there is a significant overlap between the different signs. That means that the agreement between radiologists is low. If the agreement between radiologists is low for these signs, then their utility is questionable. Here is where our project begins and what we are working on. In the radiology department, we created what we call the Dream Hub, which stands out for 3D printing, AI, and machine learning. This group is formed by physicists, AI scientists, medical doctors, students, and radiologists that work together to solve challenging problems. Regarding the hematoma expansion project, we asked ourselves if we could put the computer to work for us and predict what hematomas will expand based on features that the computers can recognize but our eyes don't. We decided to start using something called radiomics. Radiomics is a quantitative method of feature extraction. While many radiological signs and to a lesser extent clinical variables depend on judgment by a clinician or radiologist, quantitative measurements can be a more robust way of interpreting a medical image. These are the steps that we need to do before putting the computer to analyze the images. First, we need to find the studies from our local database, export the images, and anonymize the data so crucial sensitive patient information is not included. After that, we take the studies and start what we call segmentation. This means delineating the hematoma on the imaging studies. Once we have all that information, we can move forward to radiomic features. We put the data into different programs and extract information that we can't see. After that, we analyze the output and apply statistical methods and discuss the findings with their team. Once we agree on the results, we train the computer with a certain number of studies. After the computer learned how to classify the patients, we run the program in another set of cases to evaluate its accuracy. As you can see, this is an opportunity to optimize patient care in the future. Once a patient arrives at the hospital, we can obtain a CT scan of the head and ask the computer to tell us the likelihood of hematoma expansion with great accuracy. And on that note, I want to thank you for listening to my presentation. This is part of the Dream Team, and I also want to thank them for their invaluable work. Again, we want to thank and acknowledge that we would not have had the opportunity to spend as much time immersed in intracranial hemorrhage research without this funding. Thank you so much.
Thank you, Paolo. Fascinating work there. Up next, we have Aoife Riley. Aoife is a PhD student in the Department of Cellular and Molecular Medicine at the University of Ottawa. Supervised by Dr. Rashmi Katari, she is part of the regenerative medicine program based at the Ottawa Hospital Research Institute. She received the Eric Poulet Center for Neuromuscular Disease Star Award, and she'll be presenting her study on using gene therapy to treat spinal muscular atrophy.
Sorry, thank you for the introduction. So my name is Aoife Riley, and today I'm going to be presenting some of my research so far over the past 2 years, which has been funded thanks to the Brain and Mind Research Institute. And my presentation is called Exploring Low-Dose Gene Therapy in Spinal Muscular Atrophy. So spinal muscular atrophy, or SMA, is a genetic motor neuron disease. So the motor neurons are the neurons which connect the spinal cord to the brain, allowing for the transduction of signals and the movement of muscles. In the case of SMA, we actually have motor neuron death, and this interrupts the signal transduction and leads to muscle atrophy. So SMA is characterized by progressive muscle weakness and difficulty breathing and feeding. So SMA is usually a disease that affects infants, and in the most severe cases, patients will succumb to the disease in the first few years of life if left untreated. So SMA is caused by the loss of the survival motor neuron 1 gene. So in an unaffected individual, we have a copy of the SMN1 gene and an identical or an almost identical copy of the SMN gene called the SMN2 gene. And while SMN1 produces functional SMN protein, which is survival motor neuron protein, the SMN2 gene produces mostly non-functional SMN protein. So in the case of an SMA patient, they'll be missing the SMN1 gene and only a small amount of SMN protein will be produced by the SMN2 gene. So therefore, SMA is a disease of insufficient levels of SMN protein. So because it is a disease caused by the loss of a gene, gene therapy is actually a viable option. And recently a gene therapy medication has come out, and it's called Zolgensma. So this medication is administered as a one-time high dose of intravenous infusion. And the mechanism behind this treatment is to replace the missing or mutated SMN1 gene. So upon intravenous infusion, the treatment is able to target cells, and it's actually introduced, the SMN cDNA is introduced using a viral vector, which is able to target cells, enter the cell, and then deposit the missing SMN gene inside a patient's cell. And then the cell is able to use its own machinery to produce the SMN protein. So there's been a lot of success so far with this treatment. Toddlers are able to reach milestones that you'd usually never reach if you had severe SMA. So for example, some toddlers are actually beginning to walk. However, there has been severe acute liver failure as well as other side effects that have arisen after treatment with Zolgensma. And this is due to the high dose of the virus that is required to treat patients when administered intravenously. So an alternate approach has been to actually deliver it intrathecally, which is directly into the spinal cord. So this is being explored right now, but some preclinical studies have shown that there's actually a toxicity that can be caused by overexpression of SMN in the central nervous system. So both routes of delivery have toxicity concerns due to the high dose of the virus that's required. So what we have looked at in our lab is to actually look at a low dose of gene therapy and to compare the benefit of a low dose of gene therapy to delivered to the central nervous system with a low dose delivered peripherally. So to explain further, what we actually did was to treat our mouse model of SMA either with a treatment to the CNS. So this is an ICV injection, which is injected directly into the ventricle of the brain. So this allows for the treatment to target the brain and the spinal cord directly. And then we compared this to a peripheral treatment, which is an IV injection. And this is injected into the facial vein. which allows the drug to be distributed through the body with the circulatory system. And we then compared the rescue effect of both of these treatments on various features of SMA that we generally see in our mouse model, which is motor neurons, survival, weight, motor function, and different peripheral tissue phenotypes. So our first result after our injection was that we looked at the SMN protein levels throughout the body to see where the virus targeted in each treatment. And we found that both treatments caused an increase of SMN protein in peripheral tissue. So this included the muscle and the liver. So we knew that both treatment routes was able to target the tissues throughout the body and not just the central nervous system. And then we found that only in our CNS treatment was the virus able to target the spinal cord and increase SMN there. So basically so far we saw that the CNS injection increased SMN in all tissues, while the peripheral injection only increased SMN in peripheral tissues. So the first things we actually observed after this was the rescue of survival. So we saw a full rescue of survival up to 60 days in both treatments. And what we usually see in our SMA mice is a rescue or is a survival up to 25 to 30 days. And we also saw a partial rescue and weight gain. So the red and black mice here are gaining weight over time, less than a control healthy mouse would, but our SMA mice usually actually start to lose weight over time and then die at this time point. And we also saw a rescue of motor function in both treatments. And just for some visuals, these are SMA and control mice, and SMA are much smaller. They aren't very mobile, and they live 25 to 30 days, as I said. And with our IV and ICV injections, so both injections, we saw very similar results. The mice were larger, more mobile, survived much longer, but they still had a few features visually of SMA, which includes small ears and tails. So the outcome here was that CNS and peripheral injection equally rescued survival, weight, and motor function. So next we wanted to look at the actual motor neurons within the spinal cord and see how each treatment was affecting them, since we saw such a promising rescue. And what you're looking at here is a cross-section of a spinal cord stained for motor neurons. So motor neurons are stained here in red. So in a healthy control mouse, we see several motor neurons, while in an SMA mouse, we see much less and smaller motor neurons. And what we saw was that with our CNS injection, we saw motor neuron protection. So a somewhat rescue compared to SMA, while with the peripheral injection, we saw no motor neuron protection. So we conclude that the CNS directed gene therapy prevented motor neuron degeneration, while the peripheral treatment had no effect on motor neuron degeneration. So to summarize what we have so far, we observed a peripheral increase of SMN in both treatments, but only a increase in the spinal cord in the CNS treatment. And then incidentally, we saw motor neuron protection in the CNS treated mice, but no motor neuron protection in the peripheral treated mice. And then we saw, interestingly, an equal partial rescue in both treatments of survival, weight, motor function, and peripheral tissues such as the liver and pancreas. So we saw that both routes of injection led to equal rescue, indicating that motor neuron protection was actually not necessary to rescue various features of SMA. So in conclusion, restoration of SMN restricted to peripheral organs can provide rescue to various features independently of motor neuron rescue. And therefore, when this applies to therapies, which should actually not be restricted to the motor neuron. And further studies will investigate a combinatorial treatment which will consist of a low dose delivered to the CNS and a low dose delivered peripherally to try to figure out the ideal way of treating these mice as safely as possible. So yeah, thank you very much. I want to thank my lab, all of my funding, and specifically the Brain and Mind Research Institute for the CNMD Star Award, which has funded the past 2 years of my research, so this entire project. So thank you very much.
Thanks for the lovely presentation, Aoife. Next up, I'm happy to introduce Anjali Patel, who recently defended her MSc in neuroscience under the Faculty of Medicine at the University of Ottawa. She was supervised by Dr. Maxime Rousseau, and just like our previous speaker, she was also a recipient of the Eric Poulain Center for Neuromuscular Disease Star Award during her graduate studies. Her research is focused on understanding protein mislocalization in ALS using an integrative proteomic approach. Take it away, Anjali.
Okay, good evening, everyone, and thank you for the introduction, Therese. So my name is Anjali Patel, and I recently completed my master's in neuroscience, studying the molecular mechanisms of ALS in the lab of Dr. Max Rousseau. And in today's talk, I'll be explaining how we study brain diseases like ALS by genetically modifying and growing cells in a dish as an experimental tool. So to begin with a bit of background information about ALS, or amyotrophic lateral sclerosis, this is a disease that occurs due to the death of cells in our body called motor neurons. Motor neurons are cells that transmit signals from our brain through the spinal cord to the rest of the body, allowing us to perform movements. And their death leads to the development of symptoms that affect movement, including difficulties with speech, swallowing, walking, eating, breathing, among others. ALS is known as a fatal disease for a number of reasons, as patients can typically die within 2 to 5 years of diagnosis, and there is currently no effective cure. As well, we have a limited understanding of the underlying causes of this disease. And so while the exact cause of ALS is unknown, researchers have shown that it is likely due to both mutations in specific genes and exposure to things in an individual's environment. And given that these two factors are different for every individual, we see a large amount of diversity that occurs within ALS patient populations. One of these differences lies within our DNA, or our genes, which is a blueprint that acts as a set of instructions that tells our cells how to make proteins and perform functions that are necessary for life. Sometimes our DNA can have abnormal changes, which are known as mutations, and these mutations give our cells the incorrect set of instructions and can lead to diseases like ALS. So to go into this a little bit deeper, proteins are things that our cells make using instructions from DNA. And they work together in different pathways to perform many different functions in order to allow us to live. And so when there are mutations in our DNA, the incorrect set of proteins are made, and as a result, our cells' normal functions are interrupted, which leads to diseases like ALS. And so the question that researchers and scientists want to answer is how changes in our DNA or mutations lead to problems in our cells which contribute to ALS. And so we don't know much about what happens between these steps or why this happens. And this is why the research that we're conducting in the lab is very important, especially towards developing a good therapy for this disease. And so how do we study ALS in the lab? Well, scientists use different models, including mice, flies, and cells. And in the Rousseau lab specifically, we use cells like the cartoonized version that I have here on this slide. As I previously mentioned, in the nucleus of the cell, which is shown here by the darker orange, is where our DNA is contained. And we can have our cells express healthy human DNA, shown here in the blue, but in the lab, we have the ability to add ALS-causing mutations to this DNA. And as such, we are able to model the DNA found in ALS patients. And so as a result, our cells express characteristics similar to those of ALS patients. And we can take these cells, we can allow them to grow and to multiply in a dish, and then we can use different techniques to study what is happening at the molecular level. For instance, we can look at our cells under a microscope and see them close up here, like with this gray image, which gives us information about the way that the cells function, grow, and divide. In addition to this, we can also use these cells to study the proteins that are contained within them. And so as an example here, I have an experiment that I did using a microscope to look at different parts of the cell. And all of these images here are actually of the same area on the dish, but they're colored to look at different things. So in red here, we have the cytoplasm, which is the cell body or the area outside of the nucleus of the cell. In blue here, we have the nucleus, which, as I mentioned before, is where the DNA of a cell is found. We are also able to color or stain specific proteins that we're interested in knowing more about, which I've lit up here in yellow. And when we overlay or merge these images together, we can get specific information about our protein of interest. For instance, we can see here that our protein is not found within the blue area or the nucleus of the cell, which gives us information about its localization and allows us to understand more about what role it may play within the cell. And as such, together, experiments like these give us a better understanding of where proteins are found and how they work together, and can shed light on pathways that are still not well understood in the field, which may be involved in ALS. And so going back to our unique population of ALS patients, we can create cell lines to model the DNA or mutations of each unique patient. And this way we can gain a better understanding of how the cells of these patients function and what may be going wrong in these specific patients, which is likely different for every individual. We can also test different therapies and drugs using these cells, as in ALS, similar to many other diseases, there is no single treatment that is a broad cure for every patient. And although we are a bit away from this, in the future, we may be able to create individual treatment plans or therapies that are personalized to better suit the needs of each patient. And so going back to the main questions posed in the field, the use of these cell models to study ALS as an experimental tool opens up many different possibilities and will let us fill in the gaps in understanding that exist in current ALS research. And so with that, I'd like to thank the support of my supervisor, Dr. Max Rousseau, as well as the members of my lab, my co-supervisor, Dr. Jenny Angsey, as well as our collaborators, my TAC committee, I'd like to highlight and express my gratitude to the funding provided by the Eric Pullen Center for Neuromuscular Disease, without whom this research could not have been conducted. Thank you all for your time. I hope you enjoyed today's talks.
Thanks, Anjali. Fantastic talk. Next, we have Kim Thériault, who is a second-year PhD student under the Faculty of Social Sciences at U Ottawa and has affiliations with the Briere Research Institute. Supervised by Dr. Vanessa Thaler, her research involves semantic processing in older adults with or without cognitive impairment. Kim received the Suraj and Kishori Lal Scholarship, and we have a quick pre-recorded video from her.
Hi everyone, my name is Kim Thériault. I'm a second-year PhD student in the Experimental Psychology program at the University of Ottawa. I am the recipient of the 2020 Saroj and Kishori Lal Family Scholarship. I am sincerely honored to have been selected as the recipient of this scholarship. Thank you so much for your generosity. This scholarship has allowed me to pursue my passion for research at the University of Ottawa and allow me to focus on my study. Thank you so much again.
Thank you, Kim, and congratulations again on your award. Lastly, we have Dr. Jason Berard. Dr. Berard is a postdoctoral fellow at the University of Ottawa's Faculty of Medicine and is supervised by Dr. Lisa Walker. His research interest is in cognitive and neuropsychological outcomes in people living with multiple sclerosis and other neurological conditions. He received the Trainee Researchers in Multiple Sclerosis Award, and we also have a short video from him.
Hi, everyone. On behalf of myself and our research group, I want to thank the Brain and Mind Research Institute, its organizers, and in particular its wonderful donors, which have generously supported our work being done looking at cognitive fatigability and multiple sclerosis. We know that cognitive fatigability presents a significant challenge for people living with MS and can impact their ability to take part in the daily activities that they enjoy. Currently, there are few interventions that have been designed that are targeted at improving this debilitating symptom of the disease. With your support, we are in the final stages of developing and implementing a behavioral intervention aimed at improving cognitive fatigability in those with MS, with the goal of improving their overall quality of life. Thank you for your continued support, without which this work and the outstanding work being done by my peers and colleagues would not be possible.
Thank you, Jason, for the video. And thank you to all of the presenters for your fantastic presentations, and congratulations again on receiving your awards. I'd now like to introduce Romy and Haley for the Q&A portion of the program.
Thank you, Teresa and Michaela, for hosting the first part of this program. And thank you to all of the speakers. It's so interesting to hear about your research or the techniques that you're using in the lab and how the donations from our generous donors are impacting the research at the Brain and Mind Institute. We are running a little bit late, but we can maybe ask 3 questions. So at the bottom of your screen, there'll be a Q&A box. So click on that and type your question into there, and we'll get to the first 3 questions that we see. So first, I'll introduce myself. I am Haley Gertzma. I'm a 4th-year PhD candidate in the Rousseau lab. We've heard a couple speakers from the Rousseau lab today. I'm in the neuroscience program with a specialization in human molecular genetics. And my project focuses on studying a preclinical model of Parkinson's disease and the role of alpha-synuclein. I'll pass it over to Rami for his introduction. Or I can start the first round of questioning. So Brian, what is your favorite part about research in general?
So I think my favorite part is how it's constantly changing. So I remember when I first started my undergrad degree, I took my first cell biology class. I was taught that EVs were essentially just a method for cells to excrete waste. And then now I'm doing my master's, and my entire project revolves around EVs and how they could be used to help treat patients. So I think the fact that it's able to evolve in such a rapid pace, and how my research can essentially contribute to that, is probably what I find the most interesting.
Thank you. I have a question here for Anjali. What other diseases can be modeled or studied using cells?
Okay, that's a good question. So the approach that we used was that we genetically modified our cells to express disease-causing mutations. And so we can apply this approach with many other diseases that also have a genetic link. For example, there's projects in our lab that study Parkinson's, but some other diseases would include Alzheimer's, Huntington's, frontotemporal dementia, cancer, and many others.
Interesting, thank you. And a final question here for Margarita. What is the most valuable skill you have learned during your studies and your research?
Hi. So I'd probably say, on a personal level, if you can tell from my presentation, I loved being able to work with the animals. And ever since I started going on the research path in undergrad, I've always wanted to have the privilege and opportunity to learn how to work with the animals. So that's definitely a big part of research, and just how it progresses and translates into humans eventually. But then also, I think you just get an appreciation for how much collaboration goes on, like the amount of work. You definitely need to have good cooperation skills, and it definitely takes a team of people to make all this happen. So yeah, lots of behind the scenes things.
Very true, very true. So that concludes our Q&A session for tonight. Thank you to everyone for attending, and I'll pass the screen over to our UOBMRI director, Dr. Ruth Black.
All right, thank you, Haley, and thanks to everyone, to all the speakers for sharing their exciting research findings and for putting together these presentations for tonight. I'd also like to thank the entire trainee committee who organized tonight's event. And on behalf of all the research and all the researchers and all our trainee, I'd again would like to thank our donors who provided this critical scholarship support. It is so much appreciated. This entire session has been recorded and will be available on our website, and it will also be sent out to people who registered for tonight's event. I'd also like to thank the University of Ottawa Brain and Mind team who organized the events for the entire week. So our manager, Natasha Hollywood, our administrative coordinator, excuse me, Candice Fortier, our science writer, Sarah Schock. And if you would like to support our trainees doing this critically important research through our scholarship programs at the University of Ottawa Brain and Mind Research Institute, please go to our website or contact us directly. Also, if you'd like to attend our future events and get news updates, go to the University of Ottawa Brain and Mind Institute website, and also follow us on Facebook and Twitter. So again, thank you all for joining us for Brain Health Awareness Week 2021, and we hope to see you next year. Have a good night.